At Revity, we develop molecular tools that explore and modulate information flow from DNA to encoded proteins. CRISPR research is powerful, but the techniques are built to work one job at a time. CRISPR interference and CRISPR activation decrease or increase gene expression respectively. CRISPR-Cas9 knocks genes out permanently. When your research demands exploring multiple genes or pathways at once, these tools quickly reach their limits. What if one experiment could do the work of many? The Dharmacon DyadGuide platform was built to change that. The DyadGuide dual-sgRNA lentivector is versatile, where customers first choose a lentivector with only the single guide RNAs or the all-in-one lentivector with both the single guide RNAs and the appropriate Cas9 enzyme. Think of DyadGuide like the pocket knife of molecular tools. The core is essentially the same, but purpose-built. Customers next pick their technology, either CRISPR interference, CRISPR activation, or editor CRISPR gene knockout. But here is where things get really interesting. What makes this first-to-market molecular multi-tool so special is that by expressing two guide RNAs at the same time, with an option for expressing the Cas effector too, researchers can target multiple distinct genes simultaneously, or target one gene with multiple guide RNAs within a single experiment using one product. For CRISPR modulation studies, two guide RNAs to the same gene boosts gene activation or repression levels beyond those seen for one guide RNA. And for CRISPR gene editing experiments, two guide RNAs enable multiple cut-based gene knockout. Customers studying signal pathways and gene interaction networks can use DyadGuide to express guide RNAs to two different genes to see what happens with their simultaneous gene knockout, increased gene activation, or gene repression. The key to the DyadGuide dual-sgRNA lentiviral platform is the engineered bidirectional mini U6 promoter that synchronizes guide RNA transcription while maintaining the expression of each multiplexed element separate. This is a critical distinction. Other vendors offer dual single guide RNA constructs that rely on competing sequential promoters, which frequently yield reduced functionality for one of the two guide RNAs. DyadGuide eliminates that issue entirely. This novel promoter is also compatible with both human and mouse cells, supporting researchers working with both systems. The DyadGuide platform is built into CRISPR-based lentivectors, the preferred delivery format for researchers working with primary cells and hard-to-transfect cell types. But this is not an upgrade. This is a new category of CRISPR tools. Whether you're working in CRISPR gene knockout, activation, or interference, DyadGuide is the only all-in-one platform on the market that co-expresses two single guide RNAs alongside the relevant Cas9 enzyme in a single construct. With a price range similar to lentivectors with one guide RNA, the DyadGuide all-in-one platform is budget-friendly and drives cost efficiency. Other vendors offer one single guide RNA per vector, and the relevant Cas9 is supplied separately. This adds complexity, variability, and time to every experiment. DyadGuide removes those barriers for cleaner data and faster results. For customers wanting streamlined expression of two single guide RNAs only, we also have DyadGuide dual-sgRNA lentivectors for editor CRISPR gene knockout and CRISPR activation or interference. The Dharmacon DyadGuide platform represents a genuine leap forward in CRISPR research capability, creating a new category for CRISPR tools. It brings together multiplex targeting, superior expression architecture, and proven lentiviral delivery into a single, purpose-built solution. DyadGuide is ideal for academic labs performing pathway analysis, gene function, and mechanistic studies, as well as biotech and pharmaceutical teams racing to validate targets. If you're ready to move beyond single-gene experiments and unlock the full potential of your CRISPR research, It's time to explore what DyadGuide can do for your lab. Reach out to our technical support team to discover how we can accelerate your next breakthrough. The most complex experiment just got a whole lot simpler.